Welcome to my video on transcription in prokaryotes. Transcription is the process of the synthesis of RNA by copying the template strand of DNA. So when transcription starts, a sigma factor bonds to RNA polymerase and changes it into its active form, which is called holoenzyme. The holoenzyme is able to recognize the promoter on DNA and will eventually bind to it. Now here we have initiation, and the holoenzyme then binds to the promoter region, which consists of the -35, the -10, and the +1 regions. The -35 and -10 regions are very important to allow proper binding to occur. When the RNA polymerase holoenzyme binds to a region, as soon as it binds, it separates the DNA into the template and the coding strand. And a side note, the template strand serves as a template for synthesis of RNA, and the coding strand is almost identical to this, except the only difference is that it has uracil bases rather than thymine bases. And again, when the template and coding strand are formed, this creates a transcription bubble. Here you see a transcription bubble, and another thing to keep in mind is that the phosphodiester bonds that you can't see in this drawing, but they start to form, which bond the nucleotides together in the RNA chain. And after this happens, the sigma factor leaves now that the RNA polymerase has started to transcribe. And again, the sigma factor is significant because it allows the promoter to be recognized by the holoenzyme so it can start to transcribe at the +1 nucleotide region. Now let's revert back a little bit, and let's say the -10 region is deleted in an enzyme that metabolizes sugar. And as you can see here, the holoenzyme wouldn't know where to bind and wouldn't be able to determine where the promoter region is, so transcription would not occur. And now we go into elongation. So this starts again when the sigma factor is released. So the RNA polymerase is able to move along the template strand in the 5' to 3' direction. When this process occurs, you're left with an RNA strand that is the same as the DNA strand, except for the fact that the T nucleotides are U nucleotides in the RNA strand. And as this process continues, the DNA is unwound ahead of the core enzyme, and then it is rewound behind it again. So now we're going to deal with Rho dependent termination. And in this slide, the almonds represent the Rho factor. So in Rho dependent termination, the RNA has a binding site for the Rho factor protein. This Rho factor protein binds towards the 5' end of the transcript and moves along towards the 3' end, and eventually uses helicase to detach the DNA-RNA chain, which results in termination. In this last visual, the clips represent the hairpin structure. To start Rho independent termination, the polymerase encounters a region that is very rich in C, G nucleotides, and because of this, the mRNA folds back on itself, which results in a hairpin structure. This hairpin leads to the polymerase stalling, and there is a resulting U8 region that is very weak interaction, which causes the core enzyme to break off and free the new transcript, hence termination. And these are the references I used. Thanks for watching.